The topic of the day's session is on vaccines. All of us are going through this massive wave of COVID-19. Touchwood, it is actually declining over last couple of months, globally as well as in India also. And we are all familiar with the vaccines. Not only us, but even the common man is also aware of what is a vaccine. So here, I'm here to talk to all the participants with a special reference to what is the basic conceptualization of a vaccine, how the vaccines were evolved, what are all the different types of vaccines, what are their advantages, what are their disadvantages, and then a couple of other issues associated with the vaccines, both viral vaccines as well as the bacterial vaccines. Well, I am from KVFSU CVA Rabies Diagnosis Laboratory. Rabies. ಕನ್ನಡದಲ್ಲಿ ನಾವು ಅದಕ್ಕೆ ಹುಚ್ಚುನಾಯಿ ರೋಗ ಅಂತ ಹೇಳ್ತೇವೆ. ನಾವು ಯಾವುದೇ ಒಂದು ಹೈಸ್ಕೂಲಿಗೆ ಅಥವಾ ಪ್ರೈಮರಿ ಸ್ಕೂಲ್ಗೆ ಹೋಗುವಂತ ಮಗುವನ್ನ ವಿಚಾರಿಸಿದ್ರು ಕೂಡ ಯಾವ್ದಪ್ಪ ಪ್ರಾಣಿಯಿಂದ ಮನುಷ್ಯಗೆ ಬರುವಂತಹದ್ದು ಪ್ರಾಣಿಜನ್ಯ ರೋಗಗಳು ಝೂನಾಟಿಕಲ್ ಇಂಪಾರ್ಟೆಂಟ್ ಡಿಸೀಸ್ ಅಂತ ಹೇಳಿದ್ರೆ ರೇಬಿಸ್ ಅಂತ ಹೇಳ್ತಾರೆ ಇನ್ವೇರಿಯಬ್ಲಿ. The KVFSU stands for Karnataka Veterinary Animal and Fisheries Sciences University. The headquarters of this government university is located at Bidar in the northern tip of our state. And CVA is the Commonwealth Veterinary Association Rabies Diagnosis Laboratory. In fact, as Sri Srinivasu, he in fact had shared that information that this is the 12th OIE reference laboratory for rabies in the world. We are so proud that we have been the part of this. But before I proceed further, I just would like to tell our participants, what is this OIE? I'm sure all the participants are aware of what is WHO. WHO actually stands for World Health Organization. In this last 2 to 3 years of COVID-19 pandemicity, almost everyone knows about WHO and what all the guidelines that WHO is actually advising as far as the diagnosis, surveillance, and control of COVID-19 across the globe is concerned. You see, WHO is the international government agency meant for the human health, whereas the equivalent international government agency for animals, livestock, birds, companion animals, or you name any animal or the bird other than the human beings. Human beings are also part of the animal kingdom. That international governmental agency is called OIE. OIE is actually standing for, or is the acronym for, Office of International Epizootis. And very recently, this Office of International Epizootis is renamed as World Organization for Animal Health to make it on par with the WHO. In fact, this is the 12th such OIE reference laboratory in the world. And since last 2 years, we have been provided the status of this reference laboratory for rabies for 18 countries. Such of those who are interested, you can just visit our lab. This is a dedicated biosafety level 2 lab to take up this animal rabies research activities. Our postgraduate students, PhD students are there. And then we are also providing the services. And we also entertain the science students, especially the PUC science students, biology students, to visit the lab, and then we can show them the rabies virus itself, rabies viral inclusions. Ultimately, we are also for the betterment of the student community, for the betterment of the scientific community. That way, if any of the teachers among the participants are interested, you are most welcome to bring your say students to this lab so that we get a chance to motivate them, trigger them. Yes. Now let us get going with the topic of the day's presentation. Before I take you through the journey of these vaccines, let me tell you something about smallpox. Everyone knows about smallpox. This was a deadly disease affecting human beings across the globe, and there was very high mortality in the human beings because of smallpox. But the Greek physicians, they actually observed that the people who survived the infection with or the disease smallpox, they never got this disease again. Invariably, the death was very high in case of a smallpox, but there was some small chunk of human population who were suffering from smallpox, but still they survived this disease. They never got this infection once again. This very important observation was actually made by the physicians, the medical doctors from Greece. And all this observation was made some 2,500 years ago. Nearly 4,700, 2,500 years ago itself, this kind of observation was made. Ultimately, what is the insight? That means becoming infected by certain diseases gives immunity, gives resistance power against that disease to that particular individual who would have suffered from that particular disease. That means subsequently the same disease could not attack that particular individual who survived that infection, who survived that disease. Since then, the concept of immunity or a disease resistance power came into the limelight. Now, what is vaccine? What is the etymological derivation of the term vaccination? Vaccination is the process by which the vaccine is administered. But what is the etymological derivation of vaccine? Vaccine was actually evolved from the term vacca, V-A-C-C-A. In Greek language, vacca means cow, hasu. You can see the picture here. But what is the relevance of this vaccine with cow? You see, in those days, so even to this day, we have one disease called cowpox affecting the cows. But how the owner of these animals, these cows or buffaloes, he or she will know that the cow is actually suffering from cowpox. So typically the pox type of lesions are induced on the teats of the mammary gland, on the udder. This is the shape of, this is the illustration of udder. And on the surface of the udder, one can see the development of such pox lesions. I request all the participants to concentrate on the cursor. I'm moving the cursor on the on this particular illustration, where we have a number of pox lesions distributed on the teats of the udder. So such of those cows which are actually suffering from the cowpox, where the source of infection of this disease cowpox to the milkmaids, such of those who were milking such cows affected with cowpox, they picked up this cowpox from such cows to themselves. So thereby, similar lesions were seen on their hands, because it was directly to milk the cow, they had to squeeze the teat literally with their hands, and then in the process physically their palm was coming in contact with these pox lesions. So the pox virus jumped from animals to human beings. But thank God, cowpox virus did not induce such a high lethality. It is not virulent to human beings. In fact, this kind of situation that resulted in that opened up a new era in the field of vaccination itself. So what Edward Jenner in those days, he observed was, such of those milkmaids or such of those individuals who were in the habit of milking from these cowpox affected cattle or buffaloes, they never suffered from smallpox. Whereas such of those who were not in the habit of milking, who were not practicing or involved in the milking process, were found to be highly susceptible to smallpox. So that was the observation made by them with a special reference to the Edward Jenner. Then what he did, he conceptualized that probably cowpox is giving cross protection to the smallpox, because those individuals, they were already exposed to the cowpox. Their immune system was exposed to the cowpox virus. This is a double-stranded DNA virus. And it triggered the immune system. And then that immune system triggered by the cowpox, in case of the milkmaids, that conferred protection against a smallpox. That observation was very well documented by the British scientist Edward Jenner. You can see his picture here. He conceptualized this. And then subsequently what he did, he also collected the cowpox lesions and then experimentally he inoculated this cowpox to human beings, the susceptible human beings, with an intention of preventing smallpox in the human population. And he was successful. He documented that. And nearly after, say, 80 to 100 years, the great scientist Louis Pasteur, I'm sure all of us have heard of Louis Pasteur. We consider Louis Pasteur as the godfather, especially in the field of rabies, because he is the person, he's a French scientist who first developed the anti-rabies vaccine and then saved the mankind from this deadly disease way back in the year 1885 itself. Joseph Mister was very badly bitten by a rabid dog. He was immunized using the anti-rabies vaccine which Louis Pasteur was developing way back in the 1885 itself. And that vaccination was done in case of Joseph Mister by Louis Pasteur precisely on 6th July. So every year on 6th July, globally, we observe, we celebrate that day as World Zoonosis Day, because that was the day when the first man was protected against the deadly disease rabies, because it was on the same day that the scientist Louis Pasteur with his colleague, he immunized Joseph Mister, who was very badly bitten by a rabies suspected dog, and he saved the mankind from that deadly disease rabies. And it was Louis Pasteur who gave the name to this component as vaccine, or the procedure vaccination was etymologically derived. Subsequently, in China, or even before that, they were in the practice of variolation. What is a variolation? This was actually a grassroots level practice where the individuals who were susceptible to smallpox, they were made to inhale the powdered, dried crust of a smallpox lesions from the recovered individuals. So they were made to inhale that. Nowadays, we speak of providing intranasal vaccination to the travelers against influenza and then a couple of other viral diseases. Similar practice was done several hundreds of years ago in China itself. That was called variolation. But this involved a lot of risk. Because this virus was still virulent, there was high possibility that succumbing to such variolation practice would have triggered not just the immune system, but would have also caused the disease. Instead of protecting them against the disease, they would have induced the disease. This is one major limitation of the variolation. But this concept was there thousands of years ago. Yeah, now coming to the smallpox, which has actually presented many advantages that made this possible. Why the smallpox vaccine could be so effectively developed, and how is that this is the first infectious disease in case of human beings that was declared eradicated from the globe. This is the first infectious disease among all the infectious diseases which was declared eradicated from the globe as far as the human population is concerned, because there are no animal reservoirs. Something like rabies. You know that for rabies, dogs are the reservoirs. Some of the wild animals are the reservoirs. But thank God, there are no such animal reservoirs for smallpox. Nowadays, in case we want to control rabies, or in case we want to save the mankind from deadly disease rabies, we have to control this deadly disease rabies in the animal reservoir, that is mostly the dogs. So nowadays, the government of India, government of Karnataka and BBMP in Bangalore, we are also joined with them. We have undertaken mass vaccination programs against rabies in case of dogs. So if we can control rabies in dogs, definitely the rabies occurrence in case of human beings will be significantly reduced. That is what I mean to say. There was no animal reservoir of smallpox. Then this smallpox vaccine, whatever was being practiced before 1978, that was a live attenuated vaccine, that was a live vaccine. Then it provided lifelong immunity. Lifelong immunity means you give this vaccine only once. Only one dose of vaccine is sufficient to provide the lifelong immunity. There is no need to go on giving the booster. Every year, there is no need to give the shots. Then subclinical cases were not there. Subclinical means clinical manifestations are not there. The virus is hiding. It is something like a silent killer. This is more dangerous. Clinical cases means we'll be able to see the symptoms. Nowadays in COVID-19, the clinical cases means severe cough will be there, then severe throat itching will be there, and then rhinitis will be there, fever will be there. They're all the clinical manifestations of the COVID-19. Imagine supposing there is some individual who is carrying this COVID-19 virus, but is not at all showing any of the clinical manifestation, and that person can be the source of infection. That is more dangerous, because such individuals are scot-free. They can keep on wandering anywhere, and thereby they can be the source of a spread of infection. But it was not there in case of a smallpox, and there was only one serotype, only one type of variola major, we call that a smallpox virus, was there. So there was no failure in the vaccination strategies. These are some of the very, very important issues why smallpox could be effectively controlled, because of the advantages that it was associated with. So with all these vaccines developed against smallpox, at the worldwide efforts, this deadly disease in the human population was eliminated as a human disease in the year 1978. Already 43 years are over, and the same concept was actually employed for other vaccines, so that the feared diseases, they become thing of a past. Now smallpox is a history for us. Now globally there is no smallpox, because it has been eliminated. Now the smallpox history and then epidemiology and all the other things, everything is there only in the text. But it's important that we have to know the saga of development of all these vaccines. Yeah, before we get into the details of vaccines, let me quickly share some information with you as far as the difference between a virus and a bacteria. This is just an illustration, not to the exact scale. So here we have a small part of the eukaryotic cell. So human beings like you, me, and then animal, they are all part of the animal kingdom, and they are actually made of the eukaryotic cell. This is just a part of the eukaryotic cell, and it can be as big as 100 micron. 1 millimeter, if you divide 1 millimeter into 1000 parts, then it is equivalent to 1000 microns. 1/10 of a millimeter is 100 microns. The biggest eukaryotic cell can be as big as 1/10 of a millimeter. This is the size of this big is the size of the eukaryotic cell. Then we have bacterial population. Cyanobacteria, one of the biggest bacteria. Then we have Staphylococcus, Streptococcus. They are all prokaryotic. E. coli. Then you know about Salmonella, that causes a typhoid in case of human beings. You know about tuberculosis, which causes a TB in case of human beings. They are all bacteria. Then compared to these bacterial sizes, the viruses are tiny structures. They are ultra microscopic, whereas these bacteria, they are all microscopic. Whatever the student microscope or light microscope our teachers are using for teaching purposes in the biology classes or in the science classes, they using those light microscopic aids, only the bacteria can be seen through some staining technique. Whereas viruses like rabies virus, then dengue virus, chikungunya virus, then SARS coronavirus 2, they're all viruses. They cannot be seen under the light microscope. So you have to have an electron microscope to visualize these viruses. And then the virus size is as small as just 20 nanometers to 250 nanometers. That is the size. 0.2 microns or 0.2 micrometer refers to, it is equivalent to 200 nanometers. The COVID-19 virus is about 150 to 200 nanometers. You cannot see the COVID-19 virus under the microscope. We have to have an electron microscope. But I'm sure all the participants have seen at least the illustration of this COVID-19 virus in the media. Now, with all this background, let us see what is a vaccine? What we mean by a vaccine? A vaccine is nothing but a component of the same pathogen, and that pathogen may be bacteria or a virus, which is capable of producing an immune response. Say, supposing typhoid is a disease which is caused by Salmonella bacteria in case of human beings. If we take Salmonella, if we purify Salmonella in the laboratory, and then if we kill that by treating it with some chemicals, and then in case we inject the same killed or inactivated Salmonella into the body of a susceptible individual or human beings, it will trigger the immune response, and in turn the immune response will confer protection against the subsequent Salmonella infection into the same person. I hope I'm pretty clear. So the vaccine, it can be a live or an inactivated substance. It can be a part of that virus or a bacteria, and that must have been derived from that disease-causing virus or a bacteria. And then it should be capable of producing the immune response. There are two major types of immune response. One is called humoral immune response, which leads to generation of the antibodies. Here you can see that. See, this illustration is for the virus. If you inactivate this virus, if you kill this virus, it dies. And then the dead form of the germ, if introduced into the human body, then that sparks the human immune response to develop these antibodies. What all these red colored white shaped molecules you are seeing, they're all antibodies. And then these antibodies in turn will attack the same germ when it infects the same individual. This is killed. This is used to inoculate into the human body system. Then the killed germ will trigger the immune system of the vaccinated individual. The antibodies or the immune response is triggered. Then subsequently, when maybe after 6 months or 1 year, when the same vaccinated individual is exposed to the wild type or the virulent or the dangerous form of the same virus, then its antibodies are ready. The antibodies are something like our Indian army. The Indian army is all the time, it is ready to combat with the enemies. The enemies in this case for the human body are the germs or the virus or bacteria, and then these antibodies or the immune response is actually comparable to the defense force. It's as simple as that. What are the aims of the vaccination program? So we have to have the vaccination programs to protect all the individuals who are at highest risk. See, for example, myself and then my team, we are at highest risk of being exposed to rabies, because almost every day we are handling the rabid animal brain samples for diagnostic purposes. We keep receiving rabid animal brain samples from all over the country, and then soon we are going to receive them from our other SARC countries, because this is a reference laboratory. So we are at the highest risk. So myself as well as my team must be well protected against rabies. So we have to have selective immunization strategies. Every year we are undergoing, say, immunization against rabies to protect ourselves. Then to control or eradicate or eliminate the disease. That's what I told you. Because of effective mass vaccination strategies, in the year 1978 itself, the first infectious disease affecting the human beings, that is a smallpox, was declared eradicated. This is a milestone in the history of immunology. And then we, the veterinarians, veterinary doctors, animal health lovers, animal health providers, we take pride in saying that rinderpest, the deadly disease affecting the large animals, especially the cattle, buffaloes, across the globe, was declared eradicated some 10 years ago. This is the first viral disease that was declared eradicated from the globe. But the disease does not infect human beings, but this infects and then causes disease in case of the bovines, the cattle. And how it was accomplished? Through the mass immunization strategies. We have already initiated such a mass immunization strategy to control rabies in case of the dog population in the Bangalore, Brahat Bangalore Mahanagarpalika jurisdiction, the BBMP jurisdiction, which comprises of 198 wards, and we are giving all the technical inputs to the BBMP, and we have already initiated this mass immunization against rabies in case of dogs in Bangalore jurisdiction. Now, currently, the large-scale vaccinations are also done in case of children for various other diseases, and because of this, on an average, about 30 lakh children or the 3 million children are actually saved. What are the properties of an ideal vaccine? Okay, it has to be immunogenic. That means it should effectively trigger the immune response, number one. It should induce the long-lasting immunity. That means, for example, smallpox vaccine. We just give one shot, that's enough. There is no need to give the boosters, repeated boosters. Then it should be safe. The vaccine itself should not cause any harm to the vaccinated individuals, and it should not induce the disease. It should not have side effects. That is what I meant. Then the vaccine should be stable in the field conditions. Stable means it should not be degraded. It should be thermostable. That means even if it is kept at room temperature, it should maintain its immunogenicity for quite a long time. Then an ideal vaccine should also be combined. What I mean by combined vaccine? I'm sure all the participants know about the DPT, the triple antigen, diphtheria, pertussis, and tetanus. So I'm sure you have undertaken immunization against a triple antigen to your children when they were young. And then what is that triple antigen? Triple antigen means there are 3 different antigens. Each antigen is nothing but a component of a pathogen which will trigger the immune response. So diphtheria is caused by Corynebacterium diphtherium, one vaccine, one bacterium, then Bordetella pertussis, and then tetanus, Clostridium tetani. 3 different disease-causing pathogens, they are mixed. So that means they are combined and included in one syringe, and then you give the shot. So that means with one shot, you are providing, you are providing immunity to 3 different pathogens. So that is called combined vaccine. Then affordable, it should be cost effective. If it's very, very expensive, we cannot undertake mass immunization for the benefit of the public. And it should be available, not that it is available only in Delhi or in America and some other city, and we are not able to have access to that. It should not be like that. It should be accessible to all. Then effective in all subjects, means irrespective of the age group. The vaccine should be suitable for elderly people, middle-aged, young, as well as children. And then it should transmit maternal protection to the fetus. That means whenever the fetus is in the womb, whenever the fetus is in the uterus of the mother itself, the mother should be vaccinated. There are certain vaccinations which are carried out during the pregnancy so that the mothers, they become resistant to the diseases. And so thereby, the maternally derived antibodies, they are passed on to the fetus even before the birth of the fetus. So that is what I meant to say. Maternal antibodies need to be triggered, and then it is to be passed on to the fetus for its production whenever the fetus is inside the. So for that purpose, even the pregnant women are also vaccinated. These are all some of the very, very important properties of an ideal vaccine. But it's really tough to have a vaccine comprising which can satisfy all these qualities. Yeah, this is just one picture how a vaccine actually works. See, this is just a layer of the skin, and here this is the vaccination being done. And can you see these green star-shaped structures? They're all the immunogens. Immunogens are nothing but the components of the pathogen or the virus or the bacteria which causes a disease and against which you would like to confer protections to the susceptible human beings. So what happens when the immunogens are injected through this needle of the syringe? Here we have 2 different types of cells. Here we have macrophages present within the skin in the circulatory. This is the blood vessel with the RBCs and the macrophages. This is B cells, which are a type of immune cells. They are all T cells. Yellow colored are T cells. blue colored are blue cells, B cells. So both B cells as well as T cells, they confer protection through various mechanisms. Now, when these macrophages, they are exposed to these pathogens or the components of the pathogens called immunogens within the needle, they engulf them, they process them. Then this processed antigen, we call this as antigen, it is actually presented to the B cells. These are the B cells. And the B cells will undergo differentiation to make plasma B cell and then memory B cell. The plasma B cells, once they are generated, because of exposure of this processed antigen from macrophages by these B cells, they will start making these what are called antibodies. Can you see these Y-shaped blue-colored antibodies? They're all produced by these plasma B cells. On the contrary, we have what are called memory B cells. These memory B cells will have these antibodies which have specificity to go and bind only to this, but they are not released in the circulatory system, in the blood. So when these antibodies are nothing but the proteins, there are globular proteins which are released into the circulatory system or released into the secretory system, they will go and bind to these pathogens. So thereby the pathogen cannot induce infection. If these antibodies are not there, then what could happen? They are on the pathogens, it will go and infect the body cells, then it is internalized, the body cells are killed. They die off. This could happen if antibodies are not there. This is with respect to the B cell immunity. Now, I told you there is another population of the lymphocytes. They're all called lymphocytes, B lymphocytes as well as T lymphocytes. The T lymphocytes, whenever they are exposed to these immunogens, which are nothing but the components of the disease-causing agents, virus or bacteria, they undergo differentiation to make three different subpopulations of lymphocytes, T cells. One is the helper CD4 cells, another is killer T cells, another is memory T cells. What is the advantage of these memory B cells and memory T cells? Says subsequently, once the vaccination is done, maybe after 1 year or so, the same vaccinated individual is exposed to the same virulent disease-causing pathogen against which the vaccination is already done some 1 year ago. Then what happens? Immediately these memory cells, they are triggered, they start secreting the antibodies, and then within a day or two, large number of antibodies are secreted, and then the virus or the bacteria is neutralized. It is inactivated, the virus. So thereby, there are no free viral particles or bacterial particles available to infect the human body cells. So that is how the human body cell is protected. Now, what is the role of these three different subsets of T cells? Say, I told you about these helper T cells. What these helper T cells they do is they start secreting certain chemicals which are known as cytokines. Here, importantly, they are also known as lymphokines. They are the chemical substances that will trigger these killer T cells to kill the infected cells within the body. That is the role. Then the memory cell will have ability to again trigger the cell-mediated immune response in the subsequent infection by the same pathogen. So these are the two important arms of the immune system. One is the humoral immune response, where the antibodies play a major role. Then we have cell-mediated immune response, where the T lymphocytes, they play the major role. This is the concept. This is how actually the vaccine works against different types of viruses or the pathogens. There are all different types of vaccines. We have live attenuated vaccine. Live attenuated means their virulence is reduced. That means when this is inoculated into the human beings, for example, I told you about a smallpox. This is capable of undergoing multiplication within the human being once it is injected, but it won't cause the disease because its virulence is drastically suppressed in the laboratory. This is live. Then we have inactivated vaccines. For example, Covaxin, I'm sure some of you have undergone this Covaxin vaccination. That is inactivated vaccine. And this is prepared by totally treating the SARS coronavirus 2 with certain chemicals. And then the virus is totally dead or it is killed in the layman's term. And subsequently, if the same virus is inoculated into the human body, it doesn't lead to disease, but it will trigger the immune response to make antibodies. And then subsequently, the vaccine actually works. Then another type is toxoids. Then we have toxoids are nothing but the toxins which have been treated, and their ability to cause the disease is drastically suppressed. But toxoid can trigger the immune system to develop antibodies against a toxin. So toxin can cause disease, but toxoid cannot. So that is another type of vaccine. Then we have heard about these, what are these subunit vaccines? Subunit vaccines are nothing but some proteins or some polysaccharides, some part of the virus or bacteria is taken out, then it is actually used as a vaccine. Then there is another type of vaccine called conjugate vaccine. These conjugate vaccines are nothing but some kind of compounds which are generated by linking the polysaccharides with the proteins. The polysaccharides derived from a particular bacteria. It is linked with some carrier proteins. I'm going to show you the illustrations in the subsequent slide. Then it is injected as a source of vaccine. Then in the recent days, we have been hearing about these nucleic acid vaccines. It can be DNA vaccine or RNA vaccine. I will talk to you with the help of an illustration in the subsequent slide, how the nucleic acid vaccine actually functions. Then you know about this mRNA vaccine against COVID-19, mRNA vaccine, which is made by Pfizer company, and that is mRNA vaccine. I will talk to you how the mRNA vaccine works in the subsequent slides. Then the recombinant vaccine. You know about Covishield. Covishield against COVID-19 is actually a recombinant vaccine. I will again talk to you about what is a recombinant vaccine with reference to Covishield or any other recombinant vaccine in the subsequent slide. So this is to know the different types of vaccine broadly. Then again, the vaccines can be grouped into three different groups: the first generation vaccine, second generation, and then the third generation vaccine. First, in the first generation vaccine, we can have the whole organism vaccines, that is conventional vaccines. Conventional vaccines are inactivated or the killed vaccine. For example, the Covaxin, it is a conventional vaccine, inactivated vaccine. Then live attenuated pathogen. I told you about a smallpox vaccine. That is a live attenuated vaccine. They are identified under whole organism vaccine. We are not going to take a small component of that, but we are going to pick the whole virus. Either you treat it with chemicals or treat it with some physical factors and then inactivate or kill it so that it doesn't undergo proliferation when it is injected into the human. Whereas it can undergo replication if you develop a live attenuated pathogen. That means live attenuated. What we mean by attenuation? We are going to suppress significantly the virulence of the disease causing pathogen. That is live attenuated pathogen. I already briefed you about what is this attenuated whole agent. We are going to suppress the disease causing ability by reducing the virulence so that it won't cause the disease, but nevertheless, it will effectively trigger the immune response of the individuals. There are certain advantages and disadvantages for attenuated as well as the inactivated vaccine. As I already told, attenuated vaccines, just a single dose is sufficient, just like a smallpox vaccine. But one major limitation of these attenuated live vaccines is that they have the potentials to revert back to the virulence and then it can cause a disease. If that particular viral vaccine undergoes some kind of a mutation, you know what is a mutation? There is some change in the nucleic acid sequences, and then it can lead to reversion of that vaccine virus, which was originally suppressed as far as its virulence is concerned, but it has regained its virulence back because its a nucleic acid has undergone some kind of mutation. Then about the inactivated whole agent vaccine, this also I've already briefed. Here the pathogen is totally killed, but the complete structure of the virus or bacteria is retained, but still it triggers the immune response. There are certain advantages of inactivated vaccine, means there is no reversion, unlike the live attenuated vaccine. But the major disadvantage is that it does not trigger the cell-mediated immune response so effectively like live attenuated vaccine. Then we have what are called the second generation vaccines, the subunit vaccines or peptide vaccine. The advantage is that you can have more purer form of these subunit or peptide vaccines. What are the subunit vaccines? As I already told, let's say this is just an illustration of the virus. You can take only the envelope protein, or you can take only the capsid protein of the virus, and then use that as a source of vaccine. You are not going to pick the complete virus here. So if you take only some components of this virus, especially the proteins, and then develop a vaccine, then it is called a subunit or a peptide vaccine. What is a toxoid? Tetanus toxoid, we know. Supposing some child falls on the ground, and then if there is an injury, then immediately we take them to the medical doctor, and then especially the tetanus toxoid tetanus vaccine is given. The tetanus vaccine is nothing but the toxoid. These toxoids are nothing but the inactivated toxins. Clostridium tetani is the pathogen. I'm sure all of you have heard of the tetanus. And here we are using the tetanus toxoid vaccines. This is about the conjugate vaccine. We develop it by linking the polysaccharide coating on the bacteria. You purify the polysaccharide coating on the bacteria and then link it with some protein, and then the molecular weight is significantly increased, and then it is known as the conjugate vaccine. But we have very few conjugate vaccines in the system. Okay, they're all some first generation vaccine, second generation vaccine, but still there is a need for development of much better vaccines. But why? Why do we need to have new or the better vaccines? It is primarily because there is a possibility of these viruses undergoing genetic variation. I'm sure all of you have heard of different types of variants of COVID-19. We know about Delta, we know about then Omicron, and then there are several mutations in the spike protein coding gene of the SARS coronavirus 2. Several genetic variations are possible, especially in case of RNA viruses. And you have heard about AIDS, acquired immunodeficiency syndrome, which totally suppresses the immune system of an individual. This is caused by human immunodeficiency virus. And why we do not have an effective vaccine against HIV? That is because this being an RNA virus can tremendously undergo mutation, leading to generation of several genetically variant viruses of HIV. So if you go for conventional vaccine, it may be little difficult to protect against this. And this is attributed to high rate of spontaneous mutations. And then vaccines are usually effective in controlling only the clinical signs. Mostly the conventional vaccines, they can control only the clinical signs. They can reduce the clinical manifestation. They can reduce the severity of the disease, but they may not be successful in preventing the infection. I'm sure you know the difference between infection and the disease. Infection means picking up some bacterial pathogen, picking up some viral infection. The virus or bacteria enters into the susceptible human being, but it doesn't cause the disease because the immune system of the individual who has picked up the infection is strong. That person can overcome the infection, and then there is no disease. Supposing somebody is already having some comorbidity, that means their immune system is not strong. Maybe they are having immunosuppressive conditions. In that case, that infection can lead to disease. Disease is the sequelae, disease is associated with the infection, and then disease occurs wherein the clinical manifestations such as severe cough can be seen. So that is disease. Disease means suffering is there. Mere infection may lead to disease, may not lead to disease, and that purely depends on the immune system of the individual. Then there is need to have better price affordability. And it's important that we still need to have efficacious vaccines, and then we need to enhance the vaccine safety. The conventional vaccines may not be safe at times. And it's important that we also need to have the third generation vaccines, which could provide much longer immune response. And hence, we are into the era of novel vaccines, the DNA vaccines. All these DNA vaccines, they are identified under the third generation vaccines. And how do we develop that? These DNA vaccines are genetically engineered vaccine, which will induce the immune response in the host against any of the disease-causing pathogens. But how do we do that? It is actually done through the recombinant DNA technology, wherein the gene of interest, So, for example, you have heard of the Covishield. Covishield is a recombinant vaccine wherein the gene coding for the spike protein is actually cloned into one virus vector, which is something like a plasmid. And then that itself is actually injected into the human being. So here the cloning of the gene of interest is to be done in the plasmid vector under the strong promoter. This is little more technical. I don't think there is need for me because of the constraint in the timing. And please remember for the timing that these DNA vaccines are generated through the genetic engineering technology. Let me quickly go through this illustration. You know the double stranded DNA structure is there. And this is one expression plasmid. Plasmid is again nothing but a double stranded DNA and which is present within the bacteria. And then generally this plasmid will carry antibiotic resistance marker. And genetically such a plasmids have been developed in the lab and they are commercially available. This is nothing but a double stranded DNA. What the scientific fraternity have done is this, suppose let's say this is a DNA, double stranded DNA of a virus, let us say a herpes virus. You have heard of herpes and you want to develop a vaccine, DNA vaccine against herpes. Then how do we go about it? You take a small portion of the double stranded DNA of that herpes virus. Herpes virus is a double stranded DNA virus. Then link that with this plasmid. Plasmid is again nothing but a small circular double stranded DNA which is genetically synthesized in the lab. And now this herpes viral DNA is cloned into the broken plasmid. Now, once it is joined, you put some enzyme to ligate the ends. Now it is completely circular. Then we need to transform that into you introduce that clone into some E. coli cell, and this is what we have here. Okay, this is the same construct. This is introduced into the E. coli. Now the E. coli is a gram-negative bacteria with its own components. Now the E. coli bacterial cells are grown in the lab along with the E. coli cells. This will also make several copies of this plasmid DNA, which is already having the herpes viral DNA. Now what we can do in the lab, we can purify this. Now we have several thousands of copies of this generated. Then you purify that, then inject that into the human body. Now what happens inside the human body? This will undergo transcription to make the messenger RNA. That messenger RNA will undergo translation to make the protein of the herpes virus. And then that protein will trigger the immune system to make the antibodies or to trigger the cell-mediated immune response. And that is how the DNA vaccines can work. And there are different delivery systems, how the DNA vaccines or even other vaccines can be delivered. So this is just an injector and then gene gun, microneedle patches, and then through electroporation. They're all just different delivery systems, how the vaccine can be delivered. And this is how the DNA vaccine actually works. So this is a pathogen. You take out a small part of the DNA, the blue one, from the complete genome of the bacteria. You introduce that into the plasmid. Plasmid is red and this is blue. It is introduced here. Then you inject this into the human body. Then what happens? Here, once it is plasmid is introduced, this is the human body cell, and it will have this construct. The plasmid with DNA against herpes virus is internalized by the human body cell. Now what happens? This will undergo transcription, translation, leading to generation of the protein of that herpes virus, these green colored triangular shaped structures. Now these herpes viral proteins are exposed onto the surface of the human body cell. But there is no disease here. It is only the expression of the protein of herpes virus on the surface of the human body. And then these proteins of herpes viral specificity, they are exposed to the immune system of the human body, and thereby the immune system is triggered. And then by subsequent infection of the same human being with the virulent disease-causing pathogenic herpes virus is there, then it will be countered, and then the human being, he or she will not suffer from the herpes viral infection. And as far as COVID-19 is concerned, as of now, to my knowledge, there are 11 or more than 11 candidates of DNA vaccines, which are already in the clinical trial. Here we have the list of advantages and disadvantages. I think the participants can go through this little later. And I'm going to share all these PPTs with the organizers, and I request the organizers to share it to all those who are interested to go through this in detail. Then we have heard of this mRNA vaccine. We have the Pfizer and Moderna, BioNTech COVID-19 vaccines are also there, mRNA vaccines. What are these mRNA vaccines? Just now I told you about the DNA vaccines. DNA, when it undergoes transcription, it will make messenger RNA. Here, what the scientific fraternity they have done is, mRNA is generated. It is a synthetically generated. They need not have to purify mRNA from the SARS coronavirus 2 to make this COVID-19 vaccine, mRNA vaccine. Genetically, this mRNA is synthesized in the lab, and then simply it is introduced. So in this picture, you can see this is a SARS coronavirus 2. They have taken out the mRNA, and then that mRNA is injected into the body. So in reality, this is not purified. Here, they have actually synthesized the mRNA. It is only for understanding purpose. Then inside the human body, what happens? The mRNA of a SARS coronavirus 2, it is internalized. This is the host cell with the cell nucleus and cell cytoplasm. Then it will undergo translation. mRNA of SARS coronavirus 2 will undergo translation, will make proteins, the spike proteins. Then these spike proteins are released. And then subsequently, the procedure remains the same, which I had already described in the previous slide. Macrophages, they engulf it, they process it. Then the processed spike protein antigen of COVID-19 is presented to the T cells. B cells are generated, T cells are generated, antibodies are generated, and then it will eliminate this SARS coronavirus 2 COVID-19 virus in the subsequent infection for the same individual. This is how the mRNA vaccine can confer protection. But there are several challenges with such RNA vaccines. They have been found to trigger the unintended immune reaction in the vaccinated individuals. And then RNA inside the body is quickly degenerated. It is very quickly broken down. That is one limitation. And then one of the major limitations why the mRNA vaccines could not be popularized in a countries, most of the countries in the Asia is they need to be frozen. So this is one major limitations of the mRNA vaccines. So then we have what are called the vector vaccines. Vector vaccines are something like the plasmid-based DNA vaccines, where you can use these plasmids or the viral vectors. Viruses, genome of the virus itself is used as a vector. You know about this Covishield vaccine. In Covishield, the vector is the chimpanzee adenovirus, and adenovirus is a double-stranded DNA virus. What scientific fraternity they have done is the double-stranded DNA virus of a chimpanzee is isolated, and then they have introduced the gene coding for the spike protein of COVID-19 virus. Then it was ligated. The similar analogy I had already explained when I was talking to you about the DNA vaccine. And then instead of plasmid, there it was plasmid in DNA vaccine. Here it is viral vector vaccine. And this is a typical example for this viral vector vaccine is the Covishield. Yeah, this is what. Then this is vaccinia virus. Supposing you want to introduce the rabies virus G protein coding gene into the vaccine virus. This is vaccinia virus. This is the recombinant plasmid, and this is representing probably the G gene of rabies virus. You have the tissue culture system, and then it will be infected with the vaccinia virus, and then it will be transfected with the recombinant plasmid containing the G protein of rabies virus. Then there is a jumping of the gene from the plasmid to the vaccinia virus. This is red here, this is black here, you see. Then you can see this red color portion, which is coding for the G protein, is integrated into the black colored genome of the vaccinia virus. That is possible provided the tissue culture cells are infected simultaneously with the vaccinia virus as well as the recombinant plasmid. And then you can use this as a source of the vectored vaccine. Yeah, this is about RNA, RNA vaccines. So here what we do is RNA is converted into complementary DNA through the reverse transcription, and then you can repeat the same procedure as described in the previous slide. Then we have what are called these marker vaccines. You see, what are marker vaccines? So at times, it is very, very difficult to differentiate whether the immune response is triggered because of natural infection or because of the vaccination. because both natural infection also triggers antibody production. Even vaccine also triggers the production of antibodies. Supposing there is an individual who is infected with COVID-19. Let us say there is yet another individual who is vaccinated against COVID-19. Both vaccinated as well as the naturally infected human beings will produce the antibodies. So if you have the conventional vaccines, we will not be able to differentiate the immune response, whether it is triggered because of natural infection or the vaccination. So as far as marker, if you develop these marker vaccines, then it is possible to differentiate. And this is known as DIVA strategy, differentiation of infected and vaccinated animals. In case of animals, in case of foot and mouth disease is a major disease which is really crippling the economy of the rural India. And that really affects the economy of the poor livestock owners. There we have started using, employing such a concepts of DIVA. We are in a position to differentiate whether the antibodies are because of natural infection, because of a disease, or because of the vaccination. This is called marker vaccines. Then now we have oral rabies vaccines. You know, we have undertaken, I told you that we have undertaken mass immunization against rabies in the free-ranging dogs in India, and our laboratory is actually the lead institution in the country for planning all these rabies control strategies in the country, as well as in Bangalore and in other parts of the country. But major reason why we are not that successful in controlling rabies in case of free-roaming dogs is catching the dogs. You see, as far as domesticated dogs are concerned, it's very easy for our veterinarian or whosoever it is to go to the house, or such pet dogs are brought to the clinics or to our veterinary hospitals or government veterinary hospitals, and then they can be immunized through injection. They can be immunized against rabies. But what about the free-roaming dogs? It's so tough to catch them. And you know, one of the mandates of ABC ARV program of our BBMP, Brihat Bengaluru Mahanagar Palike, is to catch these free-ranging dogs or free-roaming dogs, the street dogs, and then immunize them against rabies, so that the occurrence of rabies in dogs should be almost eliminated in the days to come. But catching the dogs is a big issue. You will have to literally chase the dogs, and then it takes a lot of manpower, it takes a lot of time, and that is a limitation of controlling rabies in free-roaming dogs. For that purpose, internationally, oral rabies vaccines have been generated. And in India, we have not been using them so far. And we are very much prevailing upon the government of India to approve the importation of oral rabies vaccine. What is done here is you introduce this oral, you introduce this rabies vaccine into some bait, maybe some muscle piece, or maybe in some egg yolk, and then make some preparations and then throw it on the street. The free-roaming dog will come and then eat it. Then once it actually chews that a sachet, the plastic sachet which is containing the rabies vaccine, that breaks, that ruptures, and then the vaccine is released into the oral cavity, and then through the mucosal immunity, the immune response is very well triggered, that can confer protection against rabies in such a free-roaming dog. This is another concept which is becoming very popular, especially to control the deadly disease rabies in the wild animals. Here you can see some pictures. In the helicopter, they're all the bait vaccines. This is one sachet containing the rabies vaccine. And these two pilots, they are actually carrying these. Through helicopters, they actually fly across the area, in the forest area. And then these bait vaccines, they are thrown from skies into the forest, where the wild carnivores and any of the susceptible jackals or wolves, any of the wild carnivore which are susceptible to rabies, they eat it and then they develop rabies. Say, recently we have reported rabies in jackals in our Haveri district in Karnataka state. And recently our laboratory, we reported occurrence of rabies in wolves in Maharashtra in Amaravati district. And likewise, there are several wild animal rabies cases which we have reported. We received the heads of these animals. Oh, maybe, I'm sorry, maybe I could have shown you those pictures. Maybe I get a chance to talk to you all once again subsequently. So oral rabies vaccine is becoming very popular across the globe. And this is how you see the oral bait vaccines are actually presented. Here you can see one raccoon. This is actually eating the bait. And this bait is actually containing such oral rabies vaccines. Yeah, there are several vaccines which are being used by the veterinarians to control various bacterial diseases, viral diseases in case of different species of animals, cats, dogs, even cattle, buffalo, sheep, goat, pigs. They're all commercially available vaccines against various viral diseases as well as bacterial diseases. We have vaccines against some of the protozoal diseases also. Then we have a plethora of vaccines to control viral infections, bacterial infections in case of poultry. Then I don't think I need to take time to talk to our participants about the COVID-19 vaccines. We have Covishield and then Covaxin, and then we have mRNA vaccines elsewhere. And here we have just the pictorial expressions of different sources of vaccines. Yes, with all these, to cut the story short in conclusion, most of the infectious diseases can be prevented by timely vaccination, but it's very, very important that our general public, they must and should cooperate with the government programs as far as control and eradication of the disease is concerned. You see, this picture shows. So here, this person is so trying to convince this individual to undergo this COVID-19 vaccine, but this person doesn't want to take the vaccine. And now and then, even in the media, we come across such a situation. People are very much reluctant to undergo vaccination against COVID-19. Likewise, similar situation is also there in our animal husbandry sector as well. There is a mass vaccination programs against foot and mouth disease. Such of those faculty who are from villages, they will know about this economically very important disease, which is really very much crippling the economy of the rural India. Foot and mouth disease. There also, the owners, they don't want their cattle or buffalo to be vaccinated against FMD because they are under the impression that ನಿಮಗೆ ವ್ಯಾಕ್ಸಿನ್ ಕೊಡಿಸ್ಬಿಟ್ರೆ ಅದಕ್ಕೆ ಹಾಲು ಉತ್ಪಾದನೆ ಕಡಿಮೆ ಆಗೋಗ್ಬಿಡುತ್ತೆ. The vaccination against FMD is likely to lead to drastic reduction in the milk production, and then because every day his family runs on the milk production itself. But it is a myth. To a certain extent, it may be there, but in the long run, his cattle is getting protected against economically very important disease such as FMD. So that way, both the livestock owners as well as the public, general public, must cooperate with the government in its commitment towards effective control and then subsequent eradication of these infectious disease. So I end my talk with this universal slogan: prevention is always better than cure. This is so much so, especially with reference to the deadly disease like rabies. There is prevention, but there is no cure. Supposing somebody comes down with the deadly disease rabies, death is 100% assured. There is no treatment for rabies. Supposing somebody actually starts showing the clinical manifestations of rabies, death is 100% assured. But that can be avoided by proper pre-exposure prophylactic vaccination. That is what we say. Somebody is actually exposed to a street dog, immediately wash the wound thoroughly for 15 minutes, and then you apply some antiseptics or 70% ethyl alcohol, or then the iodine, and then go to the medical doctor, undertake vaccination. And then if there is a oozing out of blood, then you will have to undergo administration with